Hey guys, welcome back to my channel. In this video, I'm going to be continuing my series where I cover an entire IB biology topic in a quick amount of time to help you guys with your revision. So last time I did topic 1, IB biology, which was about cells. Now we're looking at molecular biology, which is topic 2. And also a thank you to Biology for Life, which is where I found this helpful resource, which is the syllabus guide. It provides you with all these unit understandings and other important stuff that you need to cover in your study. So firstly, we're zooming in. Now we're going from cells and we're zooming in further to the molecules. Molecular biology explains living processes in terms of chemical substances involved. The molecules that are interacting with each other is how the living cells actually function. So we're going to look at some zoomed in versions of um, cellular activities. Carbon atoms can form covalent bonds, so we call that tetravalency sometimes, and that means that since many other than silicon, I guess, many other atoms cannot form four covalent bonds. So what happens is because of this ability to form so many bonds, carbon can become many different things. So that's why they have a diversity of stable compounds. Life is based on carbon compounds, including carbohydrates, lipids, proteins, and nucleic acids. So these are the four main types of biological molecules, and as the name suggests, you need to know what they're made out of. So lipids are going to be carbon and hydrogen and sometimes oxygen as well. Carbohydrates, CHO as well, and nucleic acid, a bit of nitrogen because of amino acids, etc. Sorry, amino acids relate to proteins here. So proteins going to need nitrogen. Metabolism is the web of all enzyme catalyzed reactions in a cell or an organism. So that's a useful definition you guys need to know. So that's how we define metabolism, and sometimes you can get two markers where you have to define them. An enzyme is a protein that catalyzes a reaction by lowering its activation energy. Without enzymes, activation energy would be too high, and enzyme processes would be too slow for our cells to actually be living. So we actually need these cells a lot, these enzymes a lot for our survival. Next, we go into anabolism, which is where it's like the building block reaction, and the catabolism is the breaking down reaction. So like kata sounds a bit rough, where anabolism sounds kind of soft. So I think of it that way. Next, you need to remember urea as a counter for vitalism. So vitalism is the thing that non-living organisms cannot living organisms can synthesize things that we cannot artificially synthesize. This was disproven by a synthesis of urea. Urea is a waste product of ammonia. It's produced by us humans, and Friedrich Wohler actually made it himself. So not by his body, like in a lab. So that disproved vitalism. Next, you need to draw diagrams of glucose, ribose, saturated fatty acids, and amino acids in general. So that's something you need to know as a skill. So glucose, 6 carbon, 5 carbon. Um, Sugar, these are the sugars, saturated fatty acids. You need to be able to draw the glycerols and the fatty acid tails. And um, sorry, sorry, that's not glycerols. That's the triglycerides. So saturated fatty acids, I can cover them in a later video. Generalized amino acids, I've made a video on this already, so make sure you guys check it out if you don't know what I'm referring to here. Identification of biochemical, um, biochemicals such as sugars, lipids, amino acids from molecular drawings. So if you guys can do S1, you can do S2. And this is another tip in your revision if you're doing some last minute revision like I do sometimes. Um, Make sure you cover the syllabus points that cover other syllabus points as well. So if you cover this point you automatically cover this point. So be efficient in your revision. Next, topic 2.2 is actually going to be for water. Next up, when we talk about water, we need to talk about its properties. So water molecules are polar, hydrogen bonds form between them. Polarity means that there's a charge separation or asymmetric charge distribution in the molecule. This is true because the hydrogens are relatively positive, whereas partially positive oxygen is partially negative, and this allows water molecules to align where the partially negative and partially positive are next to each other. Attraction, boom, we have the hydrogen bond. Hydrogen bond is really strong, and um, that helps explain cohesive, adhesive, thermal, solvent properties of water. Cohesive adhesive, we can look at that in plant biology. Thermal properties means it's high heat of vaporization. So water compared with methane requires a lot more energy to form a gas because the hydrogen bonds are present in water, but not methane, and so the hydrogen bonds need to be overcome. Requires more energy to overcome these hydrogen bonds. More heat is needed. So thermal property. Use of water as a coolant in sweat. That's because of its thermal properties as well. Because when water evaporates, it requires a lot of energy as heat, so it takes away a lot of heat. So when you sweat, you actually lose some of your body heat, which is really helpful in a hot day. Substances can be hydrophilic or hydrophobic. Hydrophilic means you like water. Hydrophobic means you don't like water. Hydrophilic substances dissolve in water, hydrophobic don't. So when we talk about our amphipathic membrane proteins, we have the hydrophilic phosphate head, which does like water, so it aligns towards water, and the hydrophobic tails, fatty acid tails, align inwards because they don't like water. Modes of transport of glucose, amino acids, cholesterol, fats, oxygen, and sodium in blood. Okay, so oxygen, sodium, and other components in blood are generally soluble in water, and that's how they're transported. But larger molecules generally need to be transported with vesicles. Cholesterols actually, I think, and fats are transported by something called a liposome, which is like phospholipids that arrange themselves in a single layer, so that inside you can have hydrophobic substances. So fats, if you didn't know, are hydrophobic and don't dissolve in water. So you can put those things in a liposome and carry them, transport them around the body. Next up, we have topic 2.3, carbohydrates and lipids. So monosaccharide monomers are linked together to form disaccharides and polysaccharides. So saccharide always means sugar. We're talking about carbohydrates, um, so it's going to be the disaccharides two sugars, polysaccharides many sugars. So polysaccharides like glucagon, sorry, glycogen. The types of fats you need to know are saturated, monounsaturated, polyunsaturated. So saturated fatty acids contain the maximum number of hydrogen, and monounsaturated means they have one double bond or one region where there could have been more hydrogens, but there isn't. Polyunsaturated means there's lots of unsaturated components. Three unsaturated fatty acids can be cis or trans isomers. So cis means they go the same way, so the hydrogens on the double bond are the same on the same side. Trans means they're on the opposite side, and what happens is trans fats are really bad for you, so that's something you need to know. Triglycerides are formed from the condensation of fatty acids and one glycerol. So as the name triglycerides suggests, you need three fatty acid parts and a glycerol, and triglycerides are actually really are closely associated with phospholipids. Instead of the glycerol, you actually have a phosphate head, so that's a quite cool fact you need to know. You don't need to know, but it helps remembering. Structure and function of cellulose and starch in plants and glycogen in humans. So cellulose is in plants and it forms part of the cell wall, provides structure, whereas glycogen in humans is more for storage of sugar, such as when you eat a heavy meal, you can have that excess glucose stored as glycogen. Scientific evidence for health risks of trans fats and saturated fatty acids. So you're going to be given graphs. So specifically in a database question where you have to analyze whether fatty acid and trans fat consumption and coronary heart disease or some other disease associated with heavy fat consumption. Lipids are more suitable for long-term storage, for long-term energy storage in humans, rather than carbohydrates. This is true because lipids tend to be less water soluble, so they're going to disrupt osmolarity less, and also lipids are more energy dense, so they're you can pack a lot more energy into just the one gram of lipids than carbs. So that's a easy way for us to the human body to store energy, even though it may not be convenient to have fat in our body. We may not like it, but that's how it is. Evaluation of evidence and methods used to obtain evidence for health claims made about lipids. So this is where you judge the validity of the claims that are made about health. So some people say trans fats are okay, some people say it's really bad, and you can argue some ways from data. So another important thing you need to remember, as I always say, correlation doesn't equal causation. That's like the one thing you'll remember for the rest of your life or in life from these topics. Use of molecular visualization software to compare cellulose, starch, and glycogen. So this is all to do with branching. So cellulose, starch, and glycogen have different levels of branching and different types of branching. So you might need to know that. That's quite difficult, actually. I would say that this topic I might color this in red because many people don't go over this topic enough or in detail. Determination of BMI using a nomogram. Nomogram just lists height and then weight, and then you can determine whether they're overweight, um, perfect, normal, underweight, etc. So you need to be able to use a nomogram, but it's quite self-explanatory. And in a question, they'll generally mention, say, "Oh, you're using this nomogram." So um, you don't need to memorize much on that. Just do a practice question before an exam. Now we're moving on to proteins, which are quite cool. Proteins are just lots of amino acids linked together, and amino acids linked together in condensation reactions where they have a peptide bond and form a dipeptide and a polypeptide. 20 different amino acids form different variations of proteins because those 20 can be arranged. Those 20 can be put in order, and order matters with proteins, so there can be a wide variety of proteins. So yeah, amino acids can be linked in any sequence, giving a huge range of possible polypeptides. So that's the point I was covering there. Because you have 20, and let's say you have four amino acids, you have 20 to the power of four different proteins that can be made just from a sequence of four amino acids. Amino acid sequence of polypeptides is coded for in the genes. That's true because the central dogma of biology is that genes and DNA are transcribed to mRNA, which is then translated to proteins, and so the order is determined by the genes. But not all the genes have to be expressed. Remember differentiation in topic 1.6 or topic 1.1. Understanding 5 is that a protein may consist of a single polypeptide or one polypeptide or more than one polypeptide linked together. This is actually something we call quaternary structure. So when we have a polypeptide, it can combine with other ones as well. For example, hemoglobin is a structure that consists of about four polypeptides, I believe, and even prosthetic groups, which is another form of quaternary cell arrangement. I'll make a video on proteins on another day where I can cover this stuff in more detail. The amino acid sequence determines the three-dimensional conformation of a protein. So the amino acid sequence is actually what we call the primary structure, but based on the sequence, we actually determine the secondary and tertiary structures. So secondary structure is the hydrogen bonding in the protein, and this is the formation of alpha helices and beta pleated sheets. More on that on a video later. And tertiary is when you have other bonds such as disulfide bridges, ionic bonding, hydrogen bonding on R groups. Sorry, R group interactions, and these could make conformation changes in the protein. But this is all based on the sequence of amino acids, because if you have leucine next to methionine, that's different from having guanine, having tyrosine next to methionine. So moving on to the next one, living organisms synthesize many different proteins with a wide range of functions. That's true. You just need to know. There's a bit of there's a mnemonic for it, which I won't say, but you need to know about structural proteins, hormonal proteins, immunoglobular proteins, transport proteins, um, sensitivity reception proteins, and metabolic proteins and enzymes. So you need to know membrane proteins and enzymes. So you can work out a mnemonic for that yourself. Every individual has a unique proteome. That's true because remember, every individual has a unique genome, and the proteome is larger than the genome because these genes, right? Just because the genes code for a particular protein, right? The sequence is not the only thing. There's actual other interactions that occur. So your proteome is gonna be larger than your genome. So Rubisco, insulin, rhodopsin, collagen, spider silk are the examples of proteins you need to remember. It's not much memorization if you use my mnemonics. So structure, you can think of collagen, and let's say membranes, you can think about something here. I think maybe not membranes, but Rubisco, you can think about enzymatic and metabolic enzymes in photosynthesis, I believe. Yeah. And rhodopsin is in the receptors of eye, so that's sensitivity. Denaturation of proteins by heat or deviation from pH. So you need to be able to draw the diagram which shows enzymatic activity against pH and heat. So when it comes to heat, higher heat actually breaks these hydrogen bonds and these conformational uh, like bonding in protein, so it changes the structure of a protein. And when it comes to enzymes and structure are really important because structure determines how enzymes interact with their substrates. So if the structure is changed, they cannot properly interact with substrates, screwing up the enzyme. Same with pH. So if there's a lower pH, there's lots of H plus, and what happens is this all this H plus actually bonds with or interacts with the negatively charged regions of the protein, thereby causing unwanted conformational changes, and thus making the protein denatured. And on the other hand, really high pH means lots of OH minus, and lots of OH minus means that they can interact with the positively charged regions, conformational changes, screwing up enzyme again. And now you need to know how to draw the peptide bond, which I have a video for, so I would have popped up the link earlier, or just search it up on my channel. 2.5 is all about enzymes. So enzymes have an active site which substrate binds to. Substrate is the reacting molecule, so that's the one that hasn't changed that occurs to it. And enzymes remain unchanged. So I don't think maybe that's not a point, but that's something you need to remember. Enzymes after catalyzing reaction are the same as before. Enzyme catalysis involves molecular motion and the collision of substrates with the active site. That's true, but for this for topic 2.5, if you do HR biology, which I assume most of you do because hardly anyone does SL, you can actually find HL. You can actually find this topic covered more in topic 8.1 in HL, where you talk more about enzymes, temperature, pH, and substrate concentration, effect rate of enzymes, enzyme activity. So, like I mentioned earlier, and if there's low substrate concentration, enzymes aren't really doing anything, so the activity is low. Enzymes are denatured for all the reasons mentioned here. And remember, substrate concentration does not denature enzymes. Immobilized enzymes are used widely in industry. You need to think about lactose-free milk. So lactose is broken down by the enzyme lactase, and some people are lactose intolerant. So what we do is we have these calcium alginate beads onto which we can put these lactase enzymes, and lactase enzymes become immobilized and unable to move. Then we pass on milk through these enzymes, and what happens is they break down lactose or lactase into glucose and galactose, leaving the milk lactose free. Next, we need to talk about enzymes that test effects of pH, substrate concentration, etc., on the activity of enzymes. Just give it a YouTube search and you'll be able to find any good experiments and experimental investigation for factor affecting enzyme activity. So you can do some more Google searches, or your school will actually do a prac on it. Many schools do that. Now we're looking at the genetic information of a cell. We have DNA and RNA, deoxyribonucleic acids, ribonucleic acids. You're going to sometimes have to distinguish between the two. And first of all, they are both contain nucleotides, not the same ones. These contain adenine, thymine, cytosine, and guanine. This one has adenine, uracil, cytosine, and guanine. So instead of thymine, there's uracil. Instead of thymine, which is in here, there's uracil here. DNA differs from RNA in the number of strands present. DNA has two strands, RNA has one strand, single-stranded, double-stranded helix. The base composition, like I said earlier, and the type of pentose. So the pentose here is deoxyribose, and this is just ribose as the pentose sugar. So the individual components are different, and this is a very popular compare and contrast question. DNA is a double helix made of two antiparallel nucleotides and linked by hydrogen bonding complementary base pairs. That's true. So hydrogen bonding is what actually holds the structure together. So unlike the peptide bond in amino acids, but here we also have something called the phosphodiester bond, which might not be in this topic, but maybe pops up later on in HL. Crick and Watson's elucidation of DNA using model making. So they had actually before Crick and Watson and I think Rosalind Franklin as well. She contributed a lot. Um, they were disputing what type of structure DNA has, but I think Franklin's experiment was able to deduce the structure. Drawing simple diagrams of structure of single nucleotides using circles. Yeah. So this is just being able to draw one. I'll make a video on this sometime soon. And if I just leave a comment to remind me. But that's just there are different methods of drawing it, but generally you try to keep it as simple as possible with the sugar being a pentagon, circle for the phosphate group, and rectangles for the um, nitrogenous bases. The nitrogenous bases are the adenine, thymine, cytosine. Now, when it comes to DNA, replication, transcription, translation are the really important part. So, replication of DNA is semi-conservative, meaning we actually keep one of the original strands when we replicate it. So, when we replicate DNA, say from one pair of original strands, so we have strand one, strand two. When we make make copies in them, the new copies will contain strand 1 and strand 2 as part of them. So that's why we call it semi-conservative. We conserve the original. Helicase is an enzyme that breaks down the double helix of DNA. Remember, the double helix is held together by hydrogen bonds. Helicase acts like scissors and cuts the hydrogen bonds. DNA polymerase links nucleotides together to form a new strand using a pre-existing strand as a template. So DNA polymerase actually adds nucleotides in the 5 prime 3 prime direction, so that's important as well. This is all coming together in topic 7.1. Transcription is synthesis of mRNA copied from DNA-based sequence by RNA polymerase. So RNA polymerase is the enzyme that actually makes mRNA from DNA. We need mRNA because DNA is stored in the nucleus, but mRNA is able to move out of the nucleus for translation, and translation is important for making enzymes or proteins. Sorry, translation is the synthesis of polypeptides on ribosomes. You remember 70S and 80S ribosomes, so this is what they do. They make polypeptides, which are just proteins. The amino acid sequence of polypeptides is determined by mRNA according to genetic code. So based on the sequence of mRNA, we actually get a specific sequence of amino acids, thus a polypeptide. Codons of 3 bases on mRNA correspond to 1 amino acid in a polypeptide. So in translation, we have a codon, which is just 3 bases on the mRNA, and what happens is they actually are detected by an anticodon on the tRNA, so that's on a ribosome's tRNA, and tRNA has an amino acid attached to it. So um, essentially what ribosomes do is they're a code reader, so they read the code on the mRNA and then give the corresponding amino acid towards it. And that's how we have the translation of the mRNA code to a protein language. Translation depends on complementary base pairing between mRNA and tRNA. That's what I said earlier about how the codons are read by the anticodons. Use of Taq polymerase to produce multiple copies in the PCR. Polymerase chain reaction is used to amplify DNA, which is just making many copies of it. Production of human insulin in bacteria is an example of universality of the genetic code allowing gene transfer. That's the idea that you can take genes from humans and for producing insulin, and you can put it in bacteria, and bacteria will produce insulin. This is really useful for humans because we don't have to kill thousands of pigs just to get a bit of insulin anymore. And insulin is really important for type 2 diabetes people. Use of a table of genetic code to deduce which codons correspond. So you'll get this table, and you just have to write down which amino acid it's there for the codon you have on mRNA. So you got to think about mRNA and then the tRNA and the amino acid. So something to keep note of. Mendelssohn style experiment is what we have to support semi-conservative model of DNA replication. What they did was they had a heavy nitrogen environment, and they had DNA in it. So what happened was all the DNA that was synthesized in that environment was heavy, but then subsequent replications were conducted in lighter nitrogen environment. What they found was the new DNA of the first generation contained half of the all contain some of the heavy nitrogen. So they were like, oh yeah, so it's conserving. And what they found was in subsequent repetitions, there were some with the some strands with the heavy nitrogen, some without. So they did some calculations, and boom, they um, found out that DNA is semi-conservative replication. Use of a table of mRNA codons and the corresponding amino acids to deduce the sequence of amino acids coded by short mRNA of a known sequence. Okay, so this one is just again like this one right here. So using this table, you got to figure it out. What amino acids are there? So sometimes they'll give you mRNA, sometimes tRNA. So don't get confused between the two. Deducing the DNA base sequence for mRNA strand. So you gotta use the DNA sequence to work out the mRNA strand. So remember, instead of thymine, you need uracil when it comes to RNA. Now we got to talk about cellular respiration, and this is a controlled release of energy from organic molecules. So cell respiration is conducted by everything living, and always remember that just because plants do photosynthesis doesn't mean we disregard their ability to do cellular respiration, because sometimes in questions they you do have to consider the ability to do cell respiration. So understanding one, cell respiration is a controlled release of energy from organic compounds. We produce ATP, adenosine triphosphate, which is the currency of energy in our bodies, where it is used to function many it is used as it is required in the functioning of many of our cells. ATP from cell respiration is immediately available as a source of energy, so it's really quick and easy to use. So sometimes you talk about an analogy people use is money in the bank and money in the wallet. So if energy was money, ATP would be money in your wallet, whereas lipids would be money in the bank. So easy to access is ATP, lipids, energy that's hard to access. Um, Anaerobic respiration gives a small yield. So anaerobic means without oxygen. So when you have glucose, which is the starting molecule of ATP, so starting molecule of cellular respiration, you get very little ATP actually produced. We need aerobic cell respiration, which requires oxygen but actually produces a lot of ATP. The use of anaerobic cell respiration in yeast to produce ethanol. So ethanol is used in alcohol, and yeast actually produce this, and plants as well produce ethanol when it comes to cellular respiration, which is quite useful for us in the production of alcohol. Lactate production in humans when anaerobic respiration is used to maximize power in muscle contraction. So lactate is what causes lactic acid accumulation and pain when we're working out. So that's what happens when we don't have enough oxygen to power our muscles. So our bodies kick into anaerobic respiration and we produce lactic acid. So that's how we get pain when we do lots of exercise. And that's also what animals do as well. Animals generally produce lactate, whereas plants produce ethanol. Analysis of results from experiments involving measurement of respiration rates in germinating seeds or invertebrates using a respirometer. So, respirometer measures how much air is being used, and it's basically it has one chamber with air, one chamber with it's like a YouTube, right? It has a like this. It's like a YouTube, and it measures the movement of water based on air consumption by an organism. So that's a whole other video on that coming up. Yeah, that's it for topic 2.8. Now, final topic two is 2.9 photosynthesis. Photosynthesis is the production of carbon compounds from inorganic molecules with the aid of sunlight. Um, Sunlight and water. So light energy is what we use. So we can't have energy created from nothing. Remember law of conservation of energy. So light energy is becoming chemical energy. Visible light has wavelengths in 400 to 700 nanometers as well. You should know that. And this is which is useful for drawing graphs of photosynthesis, which we'll talk about later. Chlorophyll absorbs red and blue light most effectively and reflects green light, which is why chlorophyll is actually green. So sometimes you need to remember, oh, chlorophyll is green. So a question might ask you, what color, what light does chlorophyll absorb? You might think green, but no, it's actually red and blue. So it leaves behind green. Oxygen is produced in photosynthesis from the photolysis of water. So when photolysis is the breakdown of something with photons or light. So photolysis of water is when light breaks down water. You can get oxygen produced from that, which is really useful. That's why we get oxygen from photosynthesis. Energy is needed to produce carbohydrates and other compounds. Yes, that's true. We actually need to go through many cycles, and this is all in topic 8.3 in IB biology. So HL. So if you're doing HL, you don't really need to focus that much on this topic. Temperature, light intensity, carbon dioxide are limiting factors on rate of photosynthesis. You need right temperature for enzymes. Light intensity needs to be high enough for chlorophyll to function optimally. Carbon dioxide is needed because it's a reactant. So the enzyme graph shapes changes the Earth's atmosphere, rocks due to photosynthesis. So if photosynthesis rates are very high, carbon from the atmosphere in the form of carbon dioxide is taken in to the biomass, so we're going to get lower atmospheric concentrations. You can do some more of your thinking on what would happen to oceans and rock deposition. Draw an absorption spectrum for chlorophyll and action spectrum for photosynthesis. I'll be making a video on this later, which is a quite cool diagram, but it's one of the hardest in IB, which bio to remember. Design an experiment to investigate factors limiting photosynthesis. This is just measuring photosynthetic rate by carbon dioxide production or product production by varying one of the factors mentioned here. Separation of photosynthetic pigments by chromatography. So that's when you have crush basically leaves, and this is the practical we did. We crush leaves and then We use paper chromatography to separate the pigments and we see where they are. Just Google a video on it, quick and easy. And other than that, that's going to be it for topic 2 in IB biology, and we managed to do it under 30 minutes. This is the longest topic in terms of number of subtopics, so this is a quite cool achievement. Before we end the video, I want to remind you guys that any form of support for my channel is appreciated. So like and subscribe and comment. And other than that, see you guys later in a future video. Enjoy.